let me give the floor to Dr. Nitschke. And let me just jump right in. So we were wondering whether alpha-glucans in the CSF could be a potential biomarker for APBD, and our rationale was quite simple. First, we know that an APBD glycogen is accumulating in the brain. Second, we know that the brain is constantly flooded by the cerebrospinal fluid. And we also know that there's lots of precedents where metabolic and pathological changes in the CNS kind of are reflected in the cerebrospinal fluid. So we hypothesized relatively simply that when brain glycogen is high, that there will also be high glycogen in whatever form in the CSF. So the first question we asked was, does glycogen leak to the CSF? So do we actually find glycogen or something like that in the CSF? And to answer that, we don't actually need patient material. We obtained some commercial CSF from male humans without disease indication. And because we really don't know in what state the glycogen-derived material is that swims around in the CSF, we defined it as anything larger than glucose. This glucose in glucan chains can be detected in a glucose assay after digestion by amyloglucosidase. So when we measured glucose in CSF before and after treatment with amyloglucosidase, we saw two things. First, that there's lots of free glucose in the CSF. Second, that amylolysis does not noticeably increase or release additional glucose from putative glucan chains. And we concluded that if there are glucan chains, their concentration must be really, really low. To be able to detect these small amounts of glucans, we had to get rid of the glucose. We achieved that by precipitation in 90% ethanol, and here you see how the glucose content goes down. With this treatment, any glucan chains longer than 7 units could be or should be retained. After lowering the content of free glucose in the CSF by more than 40,000-fold, for the first time we saw a portion of glucose that was released upon treatment by amyloglucosidase. And when we spiked the CSF with some commercial glucan chains before the precipitation, this delta even increased, showing us that if there are glucans in the CSF, we can retain and detect them. All the results that I showed you just before this slide were obtained using a relatively large amount of pooled CSF sample. Now we asked whether we can measure the glucan in individual CSF samples with starting materials less than 1 milliliter. And this is the procedure we devised. We use heat inactivation as a precaution to prevent degradation of the glucan material by amylases that are in the CSF. Then we concentrate and precipitate, and after that, we detect glucans with an ATP depletion assay that is up to 3 orders of magnitude more sensitive than conventional enzymatic glucose assays. Here you see the quantification of free glucose and the glucan-derived delta after amyloglucosidase treatment with or without an internal standard of glycogen. So the blue delta here is what actually counts as glucan, glucosyl residues. This glucan delta we measured in CSF samples from 9 individuals, and answering the above question, does glycogen leak into CSF? With yes, glycogen-derived glucan material leaks into the CSF. Next, we asked whether CSF glucan is increased in APBD. We obtained CSF from one 64-year-old APBD patient and 8 age-matched non-APBD controls. And the CSF glucan content in the APBD patient is far outside the normal range. While, of course, our simple statistical tests corroborate this interpretation, we still have to keep in mind that this is based on one patient. And so the question, is CSF glucan increased in APBD? We have to answer it with possibly, right? We need to analyze more samples. So, of course, Birch and I are would like to thank the Research Foundation for the opportunity to talk here. And of course, there's lots of people that have been involved in the work that Berge and I have been doing. And of course, we're ready for any questions.